Hello, good afternoon. My name is Ricardo Pires. I'm here together with Luís Faria to present ASPiRe. We are a biotech company providing solutions for the pharmaceutical industry. And we are particularly focused on biological pharmaceuticals that we can simply call biologics. Well, biologics are medicines produced in living cells, and most of them are proteins. And in ASPiRe, we are experts in proteins and in protein-based technologies. Two good examples that are commercially available, so you should be familiar, or might be familiar with, are insulin and Avastin. These are two biologics that you might be familiar with. Now, if you look at the biologics market, leading the biologics market, and here represented in green, is the segment of the so-called monoclonal antibodies that from now on I'll be just shortly naming mAbs. And mAbs are interesting. They are proteins that are able to bind specifically to things. So, for instance, you can bind specifically to a cancer cell and deliver a drug to kill that cancer cell. So it's not by chance that 5 of the top 10 selling drugs are actually mAbs and represent total sales globally of $43 billion in 2010. Now, we became interested in understanding the mAbs industry, so we contacted several companies producing mAbs, and we came across this common problem that has to do with purification of mAbs. Irina Ramos, a downstream scientist in MedImmune, AstraZeneca, actually stressed in the conversation with us, stressed the fact that mAbs fail to reach clinical trials because of safety issues. And these safety issues have nothing more than to do with contaminants. Before I explain you the problems of the contaminants in mAbs production, let me go through the pipeline of developing a new drug and putting it into the market. You can divide it in 3 stages: an R&D stage, an approval stage, and then mass production to put a drug in the market. So most of the drugs that you know are actually failing during the approval phase. But mAbs, well, mAbs are different. They actually fail before, in the R&D phase, because of these problems with contaminants. And after, even for some of these biologic drugs that go through the pipeline and get into the market, contaminants are still a problem. Let me give you this example of Amantix. This is a biologic used to treat anemia, and adverse reactions because of contaminants present in Amantix actually lead to 19 patients to suffer severe allergic reactions, and 3 of them died. ASPiRe mission is actually to guarantee that this does not happen, and we have innovative solutions to clean up these contaminants. Now, before I show you our solution, let me show you very briefly how you produce a mAb. So mAbs are proteins. They are produced by cells, and these cells have to be cultivated in large tanks of huge amounts of liters, and they produce a very small amount of mAbs that you have to purify out of this, let's say, this massive soup. So you can go through a purification process that you can divide in 3 stages: a capture stage that gets rid of 90% of the contaminants, then a polishing stage. It's a series of purification steps that improve the grade of the purity of the antibody, and then a last filtration step to get your pure mAb. Now, if we look at these polishing steps, and at this stage, current solutions out there doing polishing are essentially chemically based. They are not specific to clean up the contaminants. So some of these contaminants actually go through the process and contaminate your mAb sample. Our solution is different. Our product, called the RCKlear, it's a protein-based reagent that is able to bind very specifically to contaminants and eliminate them very effectively from the mAb sample. So you'll get a contaminant-free mAb that is safer. And of course, because of this uniqueness of the technology, it's a fully patentable process. Now let me go back to the pipeline that I just showed you, just to tell you that with RCKlear, we will improve the success rate at the development stage. And let me invite Luís to follow up and present some nice aspects of the solution.
Thank you, Ricardo. So Ricardo just told you about the first key value driver that you want to bring into the market, which is improving the success rate of the R&D of our customers. But if we'll be present on the R&D phase, the subsequent approval steps will freeze us into the production process. So this means that necessarily we'll also be present on the mass production phase. So it is very important that we understand the impact of this, namely in what concerns to production costs. this is especially important because purification actually represents 70% of the total production costs. And the polishing part gets a very significant share of those costs. Well, actually, RCKlear solution enables a cost reduction in the polishing phases of up until 8%. So this is the second value driver that we want to bring to our customers. It is reducing production costs. So let us look now into the market. Today, 13 tons of MABs are produced to support the currently marketed MAB biologics. But this is expected to grow at rates above 12%, which means that by 2020, more than 30 tons will be produced, representing total production costs above 6 billion euros. So this is a huge opportunity that we don't want to miss. So let me tell you how we want to tackle that opportunity. So the first key step for us is to develop on a key strategic partnership with one of the biggest players already in that market, with whom we've already been talking to. Then we want to leverage on the brand awareness created by this key strategic partnership to conquer at least 2 additional customers within the first 5 years of operation, and with that get at least 5% share of the total MAB production. With this, we'll be looking at potential revenues of 57 million euros by 2020, turning into EBITs of around 12 million. So wrapping up, the protein-based nature of our solution makes it more versatile, while at the same time more specific, which improves the overall purification efficiency. So we want to use that to improve the R&D success rate of new MABs, to improve the safety of the biologics produced, and do all that while reducing production costs. So to do this, we will need 2 different investment phases. A first, smaller one of 500,000 euros to set up the environment that we need to support the key strategic partnership that I've told you about before. Then we'll need a second, larger phase. This means setting up the manufacturing facilities that we'll need to support us in addressing the mass production market. This will represent a total investment of 8.1 million. So, summarizing, we will be needing a total investment of 8.6 million euros, and with that, we'll be looking at potential revenues of 57 million euros, and with internal rates of return of 39%, we'll have a payback period below 5 years. This is the ASPiRe team, Biosolutions of Tomorrow. Thank you for your attention.